So I bet you can see my slide cover right now. If I change it to this one, do you see my browser?
We see your brother.
Cool. Seems I can change it just fine. Yeah. I think we can get started and move forward. Um, Thanks everyone for sticking around. So we're gonna change a little bit from the computational part to something more. Sorry, there's a noise outside. There's a big car moving by. It's gonna be more about like how do we deal with the DNA language? Like since it's not the language that we've been talking and speaking every day, there must be a way that we interrogate or like talk with these DNA or RNA or protein using some specific like tools. So like just want to add on and say hi to like those in Southeast Asia. I'm from Thailand. That's why I put the viral hippo onto my slide just to clarify that. And just to move forward right away, I think today we will talk a lot on Benchling, which is a very powerful tools for the DNA reading and also writing and editing. So just to get started, I just want to clarify a little bit like why do we need these tools? The thing is that like in the past, in 90s or like early 2000s, when we work with these DNA, most of the time we just use like Notepad, like or software, or we even like write on the notebook, like what our DNA looks like. But as Lewin said, we are having much bigger data every day. So doing that on the regular format may not be the perfect way of doing it. So there are some other software that could be appropriate for using these tools. Something that would be specific to the molecular biology operation, like having primers, having PCR fragments, which you may have heard about these words from the previous bootcamp session. These are something that were designed to be working very well on Benchling. And something that you can draw on a paper, but might not be that easy to do it in the regular word processor. You can also do that on these like specific tools. And we'll also discuss on the cloning, like based on the computer level, just to see how we can move things around and like predict what is going to happen using these tools that was provided. So here, I just want to put a background a little bit that like most of our engineering that will occur, we'll start from the DNA level that then can transcribe into RNA and translate into protein. So when we do engineering, we just focus on the DNA. And most of the time in synthetic biology, we will start from the plasmid level first. And plasmid is a small, relatively small circular DNA. So think about it as like, if the bacteria has a big chromosome, this one is another circular DNA inside the bacteria that you can modify them easier because we don't have to like rely a lot on the chromosome. So the other angle that we can look at DNA as is like, instead of the full circle of DNA piece, we can chop them off into smaller pieces. And if we try to change like each part, like a Lego, the engineering effort for these processes can be a lot easier and could be a lot more simplified when we work on it. So we'll get back to this kind of approach a little bit later when we learn more about benchling. But one thing I want to like add on to these processes, which might be mentioned in the previous session already, but again, I want to clarify that like there are like the plasmid that we're going to be looking at today mostly will be a circular DNA, relatively small. And there are like a few things that is required to make sure that these plasmids are still functioning. All the plasmid that we are going to be using will require the part called ORI or the original replication, which is required for the host to recognize this type of DNA so that they keep multiply this plasmid when the cell keep growing. If it lacks the original replication, then the DNA that get into the cell will just stay there and didn't get replicate and didn't go into like the daughter cells when they just keep growing. The other thing that would be very important is a selection marker. So if you try to like make sure that your microbe that you're engineering still have the gene of interest that you put in, you have to make sure that you can select for the variants that still have these plasmids. So the selectable marker is very important. Most of the time we use antibiotic as a selection marker. Sometimes we use auxotrophic, like let's say we have some microbe that we engineer them to not be able to eat a certain chemicals or not being able to like make a certain chemicals that were very important, for example, amino acids or nucleic acids. They will need this marker to help them grow further. So most of the time, we just work with on these plasmid rather than the chromosome, because putting plasmid into bacteria is relatively simple compared to the other processes. So these are the some kind of like simple figure I was trying to draw. So most of the time, when you try to put the DNA into microbe, we use either the chemical transformation or electroporation. According to this figure, what is going on is that they're disrupting the process is disrupting the cell membrane of bacteria so that these circular DNA can get close or maybe slip in to the bacterial cell themselves, either using like change of temperature or use a high voltage. Sometimes, when these processes, the heat shock or electric shock doesn't work well, we have to use conjugation, and that's like another possible process. These are the processes that bacteria has been using all the time in nature so that they share the good genetic information or the good materials for their growth and other things. So with that, since now I hope everyone appreciate the power of DNA so far in our bootcamp sessions. I want to jump into this very technical and very specific details on like how do we visualize or how do we read or look at these DNA features. Most of the time, when you get into the internet and look for the DNA data, they will come in different kind of format. There'll be this kind of FASTA that has like a very simple format that has like only a line, similar to like when you are writing, you have just this kind of DNA. You can open this kind of file in the notepad. That's pretty simple to do so. You can also, download a GenBank file, in which this GenBank file will have more structure. They will be organized in a way that like they have the locus, they have whatever specific keywords that are very particular. But technically for us as human, like, when you go through these kind of like structured information, it's not that simple to pass. It's not that easy for us to see what is the part that correspond to the original replication, what is the part that correspond to the selection marker. So using these simple text editing tools are not that easy. In molecular biology, there are some tools that were finally made for just reading these DNA data. So the simplest thing that you can see right here from the software called SnapGene is that instead of reading these DNA or the FASTA file as a simple, from left to right information, they actually add another strand of the DNA information onto this system as well. So when you see this, I'm not sure if you can see my cursor, but if you look at the SnapGene figure right here, you can see that they have two strands corresponding to the DNA, that they have like the top strand and the bottom strand that they match together, and they are not just like the same kind of DNA code, but they are actually the reverse complement of each other. The other thing that is very important for using these tools to visualize DNA is that like sometimes when the DNA come in the circular form, most of the text data that we have in our regular routine life is actually reading from left to right or right to left in a linear way. But if our DNA is circular, we need these kind of tools to make sure that we're still treating this in a circular way. So these are a pretty important feature that we are working on. Benchling is the tools that we're going to focus today because it's free for academic users. Potentially, it should be free for you guys to use as well. Let me see if I can share the link to the registration, just in case you have time right now. You can take a look at that. And the capability of benchling is like very similar to like the other powerful tools to do these DNA reading. They also have the ability to integrate like another text editor, like just like Google Doc or Word, that you can take notes for like everyday routine science as well. So that's something that we're going to move on and look further into our system. The other very important feature of the DNA tools reading or editing process is that like when we work with DNA, we often want to read them physically to see whether the editing that we're doing or the sequence we're synthesizing are good or correct or not. There are a lot of there are a lot of gene sequencing that has been in the market so far. I would like to point out three distinct processes, such as like single sequencing. Next gen sequencing was something that was like more from something that was based on the shotgun or Illumina sequencing, or the third generation that we usually call that for the long read. Mostly the nanopore system, they give us a lot of read, like up to like maybe 1,000 of base when the DNA pass through small pore. So this is a snapshot that you can see that like these kind of, we call them chromatogram, but there looks like the mountain that represent each kind of signal. So you see that these black mountain right here will represent G, and the blue one represent T. Sorry, the blue one represents C, and the red one represent T, and the green one is A. So with these kind of mountains like swapping all over the other, we can see or confirm whether our DNA is exactly what we are looking at or not. So most of the time, the most powerful thing you'll be using in these tools are to compare the DNA from one with the other. And with that, again, I'm proposing these two because like they're free for us user. Benchling is something that you can look into. Snapgene also provides Snapgene Viewer, which is not that powerful to the regular Snapgene, but they have like the free version so that you can download any DNA and just open from your desktop pretty simply. So the reason I want to emphasize the Snapgene Viewer as well is because like if you're somewhere like traveling and don't have like the internet, you may not be able to use Benchling because it's based on cloud. All right, so I actually generate a Benchling project so that we can look at and also like work on some project that we can have some DNA that I construct so that I can show you how do I manipulate or like modify the DNA that we have. But before we move further, I want to show you some simple thing that I think you have heard in a course of our bootcamp session several times already. Anyone recognize this figure? Like the shape of this, I think it's an X-ray image. It's from Wikipedia. If you search for this particular thing, this thing is gonna show up. You can probably guess it's actually the bacteriophage. So in our process, we have the phage engineering as well, and this is the the artwork where we try to draw them further. They have the protein part, they have the DNA part, and the interesting thing for these bacteriophage, I think this one is the lambda one, is that they have the circular genome in the size of around like 45 kb. I could be wrong, but yeah, these are pretty similar to the plasmid. They're just like a little bit bigger. So technically, if you take these lambda DNA and do some restriction digest, like take the enzyme and cut at a specific site, what you can see at the end is that like you have like different pattern of these digestion. The other thing that you can do when you have these in silico or like benchling or like your DNA editing is that like you can try to cut the lambda DNA and give this kind of pattern as well. So let's try to get into the benchling and see if we can do that. So this is benchling. I logged in already, so it's going to look a little bit different from what you have seen. You can try to generate new folder, new protocol, new whatever. I'm gonna try new DNA first. Just gonna type new, and you have to set them whether they are circular or linear. This is very important when you work with the DNA editing processes. So this is like, let's say we don't have anything, we're trying to type something down. If I type HTGAA, you see that it can get in, and it's automatically generate the bottom strand. But you can't just type, let's say I'll try to type E. It doesn't go. If I type A, it can get in. If it's not a nucleic acid, it doesn't work. But H is sort of like an extended like nucleic acid that can be put in here because H sort of like represent not G. That's why like H and D can also appear here as well. So If you just keep like, typing something random, you see that like not everything will come. You see that like if you have to try and like type everything down into this process, it's gonna take very long. So let's go to Google and try to search for lambda DNA. Page lambda. So there's a lot of like results that coming up. If you go back into the database that we have been touching on the previous session, you see that like something from NIH NCBI is very trustworthy for us to look into this information. So right here is the genome, complete genome of phage lambda. And if you want to take this information to open onto your computer or in Benchling, we can actually download them. Like if you click here, file in GenBank, you can create file and they'll be downloaded. But for Benchling, since it's a cloud process, you can actually import the DNA sequence and you can import from database and type it down here, search, it should show up. Then this is the name that it was given by the database. You try to import and it should pop up. Let me check the chat real quick to see if we have any big question. All right, it seems like we're doing good. Okay, back to our DNA. You can see that our DNA, we have like source right here. This is a very general thing for when you get the DNA from other online source, they'll just like label these as a source right away. You see that like if you read HTGAA sequence here, it's very hard for our human brain to keep track of what is going on. So the annotation is a very important thing to lead us further on like what is the meaning of this DNA. So let's try the plan that we were discussing about of like, trying to do the simplest thing, which is just cut this DNA. And you see this again in one of our lab on the DNA gel art. I think the information that is available right there is that using Hindi 3, I hope you can see my screen when I hop around. This Hindi 3 said that like, it's gonna show 1, 2, 3, 4, 5, 6 bands show up in Hindi 3. I'm gonna come back and type Hindi 3 here. And they show 6 cut sites. So theoretically, if you cut the DNA 6 times on the circular DNA, you get 6 products. And the size of them will show up right here in Benchling. Again, it's very different. If you cut the linear 6 times, you get 7 parts, but you cut circular one, the first time you just get the single part that was linearized. So this is another important thing when people start working in biology that may not recognize right away. If you run these digest, these are the part that they showed. If you click right here, they're gonna highlight what is available in this linear map. Keep in mind, you can move these section around. You can do some clicking and they will just highlight the part that is more important to you. One good thing about Benchling is that they give you the virtual gel. So this is going to be very important when you're working in the lab, because if you're trying to make a new DNA and you're trying to isolate them in the agarose gel, which we mentioned in our class, you have to know what is the size. And this is, a very simple tools that can show you what is the expected size that you will be looking in the lab. So as you can see here, they have 1, 2, 3, 4, 5, 6 parts. They actually have another part right here, which is pretty small and not show up in this cut. So sometime when you run the agarose gel, if you run too long, there's something that is very small like this like 500 base may not show up. So that's probably why they don't put it there in their website. So hopefully like you can see like the power of Benchling briefly when we're trying to interrogate the language of DNA. The other example that I want to show you guys is that like if you try to make new plasmid, like engineer the plasmid, which is something we are going to do all the time if you're a synthetic biologist, we would do some kind of like cloning. Or the full name of these processes is molecular cloning. We're just like doing copy one thing to the other thing at the molecular level. when I first hear about the cloning, I keep thinking about the chip that was cloned, you know, that they were genetically identical. But this is like something at the molecular level, and technically they look the same because you keep making the same plasmid over and over. So that's kind of like the reason why we call them cloning, I believe. In this slide, if you go and download the slide, you see this hyperlink that you can take a look into this sequence. So what I'm trying to show you here is that like with this plasmid, can we do some modification, like do the restriction digest or like alter some part by chemical synthesis and just make different DNA so that we can combine new type of DNA and make new type of plasmid together. So here, if you want to make this plasmid that has three things, they should have the original replication. They should have the antibiotic resistant gene, which is ampicillin resistant gene right here, and the GFP, so that they have like the green color when you shine light on it. We will try to connect these three parts together. So these are the highlight of like how I designed them in a way that they work for either like overlapping Gibson assembly that was mentioned, and also like the restriction digest way of cloning. In this case, I'm going to show you in the restriction digest way. Like let's say that we PCR or other gene and we try to just cut them with these three unique enzymes. You see SGS1 right here, KFL1 or box1 right here. These are something that we're going to use to cut these genes into the unique site so that they have like the sticky end that they can join and these Lego can combine together into a single plasmid. So these are probably like, you know, simple enough to see how do we link them together, but it's going to be a lot clearer when we go into Benchling and show you what actually is going on right there. All right. In this folder, in this project, I actually organized them into ligation, Gibson, gRNA, and Golden Gate. So in this part, we're going to work on the, the ligation first. So these are some part that I will generate them either using PCR or use like gene synthesis. One thing that I kind of want to show you guys is that if you have some DNA right here, that you have some primers. These are the process of the smaller DNA that we design and just like label them with this kind of like arrow, like half arrow. We call them primer. So these are a single-stranded DNA that we can use them to make PCR. So for example, if we take this primer that bind to around KFL1 site and the primer that bind around this FSE1 site and link them together, you can technically generate the PCR products, just like I'll show you again. You click this primer and also click this primer. You go to the right-hand side, which is the primer button. And if you go to pair primer, you can pair them and you can create a PCR product. You have to choose where you put them in, similar to like when we generate a new Word document, and this is the new fragment you generate. Technically, this will be almost identical to one of the fragment that we're making, which is this like MR part. With this part available, I'm gonna go down to these assembly tools and try to do digest and ligate. So what I'm trying to do here is that I'm gonna start with the 1A part, use it as a backbone. They'll ask you to select the restriction enzyme as well as part of the sticky end or like a blunt end generation. You see the ending showing here. We have part 2 as an insert. Again, it was kind of highlight here if you try to use that enzyme already. So it's SMI1 and FSE1. We're gonna set this. You see that like this is not going to be successful because the overhang doesn't match up. Technically, that's correct because we need the third part, which is the COI one, the origin part. If you go here and select box 1 and SGS1, it's not going to work because the enzyme cut site doesn't match. So if I redo it again, reset, and do FSE1 and SGS1 instead. It's gonna be correct. They're just like, it just show us like there's no error. Looks like everything checks out. If you click yes right here, done, give them some name right here. They'll generate a new file for you to show you the 3 origin of these things, and also like have some information about this kind of assembly that you're doing in this in silico process. So there's a question right in the chat that like, can you also use Benchling to design primers? The quick answer is yes. So if you just do a quick, you know, dragging, I hope you can see like the bottom part of my screen. Let's say I'm scrolling through this part of DNA right now. There will be part that called length that show me how long the selection is and what is the GC content, what is the melting temperature. Let's say I want to generate primer. I can right click and do the create primer, and you can just name them primer 1. This is the process of like making the in silico primer, and all of these data will be collected in your project in your folder. So I hope that answered your question like briefly. All right. Now I think I'm going very slow, but hopefully that helps you guys like, you know, get into the details of what's going on. Technically, when you do the cloning and you start with something that doesn't have the GFP, let's say we start with something that has only the covalent part but no GFP, we take that part out and we try to combine this part together. You will see that like we can change the bacteria that has no color into green color, and we grow them up in the liquid culture. You see the color difference pretty clearly as well. In the lab, it's going to be a lot clearer if you spin them down, like, you know, pack the bacteria together. You see that this pellet is very green, even though we don't shine any light onto them. The white light is like surrounding the lab can actually show you the color already. So what if we want to do another technique where we not just like, you know, put things together, but we try to change something that's already there to something else. In your case, when you do it throughout the class, it's going to be changing, you know, let's say GFP to any protein of interest. But in this case, let's say I'm interested in a protein that gives a different color. We're going to have that in the Gibson lab as well. We'll try to do the Gibson assembly here in silico, where I'm going to try to generate this fragment and take this fragment from DNA synthesis, let's say, and assemble them into a new plasmid that change from the green color to the red color. So again, we can jump back to Benchling. All right. Back to Benchling here. On the Gibson side, we will have this, we will actually use this CoE1 GFP, this plasmid right here. Again, if you have too many things, you want to close the tab, just right click and you can close other tabs. We're going to start from this one and try to change the GFP part to the RFP part. So if I go back to my notes, you see that I try to digest them around maybe like PAC1, MSS1 around there. So let's do the digest. If we can go here and select our digestion enzymes. And I think what I plan, I'm gonna cheat a bit because I have tried that already. This is what I cut. So if I want to know what I have done previously, I can sort of like scroll around here, hit Control-C, copy that part, and search into this thing and see what have I cut. So from what I do the search right here, it seems like I was using MSS1 to do the cutting. So I'm going to type in MSS1 right here. And the other part is probably PAC1. If I hit run digest, it's similar to when we do the lambda DNA digestion. They will show us two parts that will be resulting from this digest like this. We want to make sure that we grab the right part, which has everything but the GFP. So we're actually grabbing this bigger size, which is around like 2.2 KB. With this, we can just copy, right click, copy the whole thing and put it into a new DNA. Let's say digestion product. Now it's supposed to be linear, and you can do Control-C, Control-V, similar to how you work in Google Doc, in Microsoft Word, in Notepad, whatever in your computer. They will be copied here alongside their annotation. So this is very useful when we do this process in this kind of tools, apart from just like word editor, because they take not just your DNA sequence, they also take your annotation and the primers that were attached, which is going to be useful in the long run. So now we have this digestion product. I'm going to show you how I want to take the MRP part from this process. So I previously decided that like this part is where I want to take the MRP from, which is this one that will have the overhang to the PAC1, and this one that will have the overhang to MS1. I can grab them and also pair them and create piece of product. We can do that right away and generate a new fragment. But let's see if we can use a Gibson assembly wizard right here to assist us with this process. So again, this is a tool that was like already inside the Benchling itself. So it's probably like very useful for you. All right, for the backbone, I'm gonna select this as a fragment right away. And for the insert, let's see, I'm gonna go here and I'm gonna select something around MRP. In the transcription and translation process, you have to make sure that you have not just the gene, but the promoter and the terminator for this reading as well. So let's say I just like grab something, ignore whatever primer I used to have, and set this as a fragment, and tell them to do assembly of IP. What we have got at the end is this plasmid assembly and also the primers that is associated with them. So I'm going to tell them to put into Gibson for all of them, create. This is what we've got from the assembly tools. They suggest us that we should use this primer and also this primer for the PCR, and use the other pair for the PCR so that we generate two fragments from those two templates to do the Gibson assembly together. So this is something that, you know, if you don't have a lot of experience designing primers, you can ask the program to help you design this kind of Gibson assembly process without trying too hard to, like, you know, read a lot about what is the annealing temperature, what is the GC content, what is the key criteria of like trying to design. There's have some sort of like automated system that can help you get this DNA sequence that can help you assemble this protein of interest or like plasmid of interest already. So hopefully that helps explain you how do we get the new plasmid when we change the green fluorescent protein to the red fluorescent protein. So here, the results of these like digestion or PCR, we'll be running them through the gel, and then we do the transformation. And finally, you see that like the green colony will change into the red one. With that, those were the simple one. And in the whole lab, you have some CRISPR tools in your processes as well. So I want to touch a little bit on the CRISPR side here. So in my PhD study, before I joined the class as a student like you guys last year, I was a PhD student in U of Washington in Seattle, and I was working on CRISPR. So I think like maybe if I can take what I've been working on and show you guys and like how I do it in the everyday life of the researcher, that could be kind of cool. So what I was doing, and when I do the CRISPR research in bacteria, is that like I have to design a new guide RNA for the CRISPR system all the time. The CRISPR system work with like at least two components. They need at least two components, which is like the CRISPR Cas protein, usually Cas9 or Cas12, and the guide RNA, which in this case represent the guide RNA. So when we want to change the CRISPR system, we usually change at this spacer sequence around 20 base right here. And it's pretty simple to change things around. Let's say, if we have some cut site design right here that we're going to cut right here just before the spacer, and right here after the terminator, or like or anywhere in between, we can technically change this sequence in a very cheap like processes by just ordering one oligonucleotide or one primer, and that should be around like $10. And you can just get new plasmid in around like maybe 3 days processes. And this is why CRISPR has sort of like revolutionized the field, because designing new things to do editing or to do like gene modification or regulation is so simple. So the primer will look like this. It would just be something that bind to the core guanine sequence. It will have some overhang to the promoter part, and we'll just change the spacer sequence right here. And with that, before we move too far, I just want to show you how I do it on Benchling. It's going to be very quick. So this one, I'm going to move a little bit quicker. I start from having these PCK411 in which I deposited on HTGAA. I can actually show you how we retrieve this thing as well. If you go to Google gene PCK411, it should show up right here. And one cool thing eventually is that you can just copy this URL right here and import the DNA from HTGAA as well. Sometimes they'll ask you if you want the HTGAA sequence or the depositor sequence. That depends on how they were deposited. Sometimes if you go to HTGAA, there will be some sequence that were uploaded here that you can just click and download and just re-upload through the same process as well. So with that, let's take a look here. One con for like taking the sequence from HTGAA is that like they may not have the original annotation that the depositor has put in. So if I come back to my my own plasmid sequence right here, you see that like this is the primer that I have been using from time to time. And I'll just change these 20 amino acid into a new primer. And in that case, I can just make like a new plasmid all the time to target new things. This is the sequence that was used to target RFP to begin with. So let's see, if I try to identify what is the cut site that would be useful, I just have to look around here. It's probably designed by some previous researcher already. And if I just want to check real quick if they're compatible, the important thing about using these restriction enzymes is that you want them to cut only at the place that you want them to cut. If they have another place that just cut randomly inside the plasmid backbone, that our design would be improper and they may not work as we planned. So I want to check that SPE1 has only one cut site. Yes, this is the number of cuts that Benchling show us. And also BSPT1 is also only a single cut. So if we do that, this is probably the fragment that I will get from running a gel. Another cool thing on Benchling is that if you're doing like some parallel tasks and you're trying to run the gel on the same gel, but like different kind of fragments, they also show this like comparison of gene fragments that show up here. So the other part I would do is that I'm going to PCR this thing up and I'm just going to combine them by the Gibson assembly or something similar. And after that, I'll just get a new plasmid where I do the transformation. So, all right, we're going to go a little bit quicker now because I've been talking too much. In this case, if I design the guanine to target this plasmid called PCK760 that has 2 reported genes, I designed this in my research where I have the medium strength promoter expressing the GFP, in which it's going to give the green looking colony like this. And it also has another weak promoter expressing red fluorescent protein, in which case, like, they should express the redness, but not that red, because if they are just so weak, they're not going to show up. So right here, if I try to do CRISPR interference or CRISPR-I by blocking the transcription, the reading of this DNA sequence will be disrupted by having the physical blocking of this, readout. Like think about you're driving a car. If there's like a big truck blocking your way, then you can't move forward and you can't see the whole road. And that's how it works on the CRISPR interference. The other way is like trying to do activation. So sorry, I forgot to say that like if you suppress the GFP expression, the color of the bacteria will technically change from green to colorless. And if you try to do, another add-on by activating these gene red fluorescent protein, you can technically turn them into the red one. This will involve like a more sophisticated design in which like it was my PhD work. But technically, if you put some switch on somewhere upstream of the promoter with the proper design rules, you can activate this gene, and that's why you can turn these bacteria from green into red, kind of like changing the traffic light. So with that, we can technically like try to redesign the tools for like try to target the genome. Let's say if we have a gene called MC, which is for ampicillin resistant gene in E. coli genome, we can technically go and search in the E. coli genome and try to find the gene that we can probably like do the CRISPR repression or the gene knockout. So let's do that real quick. I'm going to jump back to Benchling and I'm going to look for the E. coli genome. Because it's gonna take a while to download the genome, I'm not gonna do the download right now. But let's see if I search for coli. U something. Sorry, can't remember. U0096. These are the gene sequence that show up that I probably have them somewhere in these processes. If I get into the genome, usually it takes like a little bit longer to download the file and try to search for the sequence. I don't really know what MC is, so I can go to NCBI and search for the DNA sequence. But the good thing about these processes that were annotated is that like I can probably just search in here, type the gene name MC, hit right there, you'll find MC right here. So the design rules for CRISPR is that like they need the 20 base that has the PAM sequence adjacent to it. And the PAM sequence is technically any NGG sequence. You see that like they have like up to 143 right here. If you don't really know how the sequence was designed and annotated, I'm just gonna show you like a very like simple engineering trick by just see how the other people has done before. For example, this is something that will target the MRFP. If I go to something that has MRFP, and just search on like how does it look. This is 20 base that will bind this MRP gene. If I search for the NGG right here, you see that like these are probably the GGG that were assigned. So again, if I try to search for the sequence, you see that GGG is right here, and that's our RNA sequence. So if it's on the bottom strand, there should be on the right-hand side of the GGG. If this NGG is on the top strand, then this sequence should be on the left-hand side. So that's how the design works for the CRISPR targeting. And if we come back here and look for NGG, we can probably do the same thing. Take these 20 base. You see 20 base right here. Copy it. Make sure that you copy the reverse complement because we're selecting something from the bottom strand, and go to our 411 right here and just try to replace them like this. Then you have a new guanine or a new plasmid that you're trying to make. You change these 20 ends into whatever sequence that you just added here, then you get the guanine that you're trying to design. If this is not what you're trying to do, and you just did that by mistake, you can revert it back to the version. So they have the version control in Benchling as well. I think that's what I want to show. With that, I think this is probably the last one. So since we have the guanine, we can also try like, multiple assembly of these guanine processes as well. So imagine that we have multiple guanine, we try to, replace or activate multiple genes at the same time. This is what I did in my PhD lab. Try to connect these 3 parts together. So I can technically do the PCR to make 3 orthogonal parts that can be recognized by the type 2S restriction enzyme, get them cut, and then try to connect them together through these sort of like Lego connection with the same color. If they connect properly, they'll be like this. They will have like 3 guanine being expressed on the same plasmid. And let's say, starting with the plasmid that looks like this, that has the GFP inside the plasmid, if we get the correct plasmid output, then they shouldn't have the GFP anymore, but have the guanine instead, so they will turn into a colorless processes. So with that, I want to show you briefly on Benchling how we do it. So here, going to the Golden Gate, if I ask the assembly wizard to help me do the Golden Gate assembly, they'll just like pop up the similar thing on which like we have the backbone, what is our insert. So in this case, I know that I'm going to have 3 inserts. I'm just going to add them right away. I have these backbone, this is the GFP dropout. I'm just gonna Control A, which is like select all for the whole thing. I'm gonna let this assembly wizard help me out by choosing where the BSA1 cut site is. If it set fragment, it actually show me what the BSA1 cut site is right away. Then I go to part 1 and I try to set the first insert. Again, I'm setting the BSA1 cut site. Seems like it's going okay. Then let's go to the second insert, do the same thing. No, this is not going to work because I did it wrong. I actually didn't check the BSA1 cut site. So this product turns out to be a blunt end. So I have to change it somehow. So come back and change the one with BSA1, and that's going to give you the sticky end. You will see the left-hand side is connected to the 4A part already. And the last one, by choosing the right one, you get the assembly that looks right, and you can assemble them right away. Let me show you how do I, if if it's incorrect, how is it gonna happen? So this one, if I tell them to use like this modified 4C fragment, it's gonna give you this error that like, hey, you can't assemble this. It's never gonna work because this full base sticky end, it doesn't match with each other. So either go back and change into the correct one, otherwise your cloning is not gonna work. And that's how we make this Golden Gate assembly. Once you assemble them, you get this kind of product that have these label of the backbone and the label of what the insert you are putting in. Maybe for the last thing that I want to show you in this Benchling process is that like they have the notebook feature. Let's say paste. And I don't know how to change this like languages, but FYI, if you're using something, if you're using the language apart from English, you can sort of use that as well. That was Thai. And the good thing that we can do here is that we can make a table and we can make something like classmate, and we can click at, which is like looking for some object in Benchling, and then ask them to look for 4 prime, 4E1. You can actually link them right here and click here to go for the actual plasmid. So this is pretty convenient in terms of organization. You can also make a shareable link and send it to your colleagues so that you can share your information pretty conveniently. So with that, I'll come back to wrap up my presentation here, which is like, Benchling is not the only tools that you can use. It's just one of the option. You can also use BioPython, similar to what Suwin and Paul has shown us previously. This is what I wrote when I was rotating with David Baker. We were trying to make some new kind of proteins, and I was very new to Golden Gates, and I don't know how to use Benchling at the time as well. So I just like struggle on how to do it, and like seems like I can try to teach myself how to code. So I just like keep writing these things, make some function. This is based on BioPython. I'm not trying to show you how it works, but like technically, they just like take these as like how to make the step 1, step 2. Like they put a new DNA into this building block, how to assemble them into the first layer and assemble them into the second layer, and that sort of worked out great. So there was like some kind of like some tips that like if you are not familiar with the tools that is new to you, you can also, if you're like have the computational background, you can go back and use the tools like BioPython to leverage your expertise in this kind of new research in the biology or synthetic biology. So the key takeaways for today is that like if you want to work on molecular biology tools that you have to read DNA, RNA, or protein, Benchling or Snapgene or the tools will be something that helps you a lot, apart from just using Google Doc or just using the notepad that people has been doing in the 90s. Again, I want to emphasize that Benchling is very simple, and doing the in silico molecular biology work is very important, because sometimes by checking that your primers or your DNA is compatible with each other, can save your time for multiple days. Because if you go and just like keep doing the lab, but your DNA are actually not compatible and you get no colonies, no bacteria ever, and it's from your design, that's kind of like a small disaster going on, because you get so confused, you get frustrated. So doing this kind of in silico work before you head into the lab save a lot of experimental time, especially when it's a challenging task. Finally, these are the resources. I believe like the information, like this updated version of slides is already on a shared Google Doc. But apart from Benchling, there are Snapgene and Snapgene Reviewer that you can use. Genius is another tool. I guess it's a paid version. Ape is a free version, but the UX UI is not as convenient in my opinion. But it's like local. It's on your computer. You don't have to get into the cloud to use them. This is their logo. Of course, there's an ape right there. They also have a paper that you can read on. So if you want to figure it, you want to explore, maybe like this is going to be your new favorite tools, you can feel free to go for it. There are some other information about molecular biology cloning you can read at this Genius guide or go to AdGene. AdGene is also making a lot of good like resources for molecular cloning or like for whatever molecular biology or any biology related works. AdGene has a lot of resources to share with everyone. They also have YouTube and find their YouTube video very resourceful. Again, this is the website for BioPython. There are some package that you may have to install before using BioPython to like take a look at right there. And the last one that I really want to emphasize is that like when you get a new plasmid or you're trying to make something new, and you don't really know what the annotation you should be making first, this is a very good web tool from Barrick Lab. I think Jeffrey Barrick is at UT Austin. It's called Planotate, coming from plasmid and annotation. So you can upload the sequence and get the annotated plasmid sequence according to the NCBI GenBank system. So these are very good tools that I want to share with you guys. And I think that's all I want to share. Hopefully that's useful for getting started. And maybe before I stop sharing the screen, I want to emphasize here that you can also work on the amino acids, and let me make something random. Again, this thing can be typed down only if they are amino acid, and they can show you, these quick analysis on the amino acid side as well. So I didn't really use a lot of them, but hopefully if you guys are going to work on the protein project, this is going to be very useful in the long run. With that, I'm going to wrap up my presentation here and give the floor to Adrian so that he can share about the accessible labs.